again, this is very, very easy to see. From the name, it's also easy to see from the structures that are sitting right there. Here, perhaps you can see. What is happening when this carbon right here gets oxidized to this carbon right here, besides the oxidation? A phosphate is added. Check this out. Now, what's important about that phosphate? Mass. Yes. That phosphate is not like any of the other phosphates that you see on here, with the exception of this guy right here. But what is that phosphate? How is that phosphate different? It's way more unstable, which means it's a much higher energy phosphate. It wants to come off much more so than the other phosphates, except for this one. And now, does anybody remember? Does this phosphate want to come off more, or this phosphate? And don't say yes. Colin?
The phosphate connected to the carbonyl.
This one right here
Yes.
is not the right answer, all right? And you are allowed to say, since we didn't have to memorize those numbers, I know that part. Does anybody remember how much more energetic this is, than this? Again, you don't have to know this number, but you might have written it down. Kaylee told me the other day that this one is worth a negative 11 kilocalories per mole. And she figured that out because I told her how much this average reaction was worth, and she kind of knew how much this reaction was worth. This is a +7. This overall is a -4, which means that this must be a -11. Does anybody remember what this guy is? Yeah, so then Emily, write it down. Just write it down on the board. I guess you probably didn't pay attention to it. Yeah, well, yeah, ask what you got.
-14.
-14, is that more energetic than a -11? Yes, it is more unstable. Fact is, that's almost worth how many ATPs? If each ATP requires 7 kilocalories to be made, your -14 should almost be able to drive the formation of 2 ATPs, correct? It does not. Where is this 2 ATPs going to become important? We're talking about the reverse reaction taking place. That's a very, very important point, all right? Did you have to memorize that number? No, Allison just saw it the other day, and she recognizes that that's pretty energetic. At least I hope she recognizes that. She did seem to recognize that part right there. Is it energetic enough to drive the formation of an ATP? Absolutely. That's exactly what happens right here, right? These are the 2 you get. Bonna says, no, you actually get 2 glyceraldehydes. Why? No, not Katie, not Colin, and not Bonna. Anybody else? What do you got, Jackson? How is that possible?
As G3 is consumed.
Okay, no, it's like you're talking about the kinetics. I like that answer right there. Tell me how you would chemically make that happen. Ooh, do I take this carbon right here, this is carbonyl, and have these 2 carbons switch places? I gotta do the hydrogens, folks. And this reaction is exactly like what other reaction? On your cheat sheets, what other reaction is this like?
Reaction 8, number 8.
8 is not correct. That is also an isomerization, but not like this one. The one we're talking about, this is exactly the same as what's happening right here, which is what?
Reaction 5.
Reaction 5. This is reaction 5. Making that into that, that's reaction 5. So yes, it's identical to itself, okay? What other reaction besides reaction 5 is identical to reaction 5? Eric?
Reaction 2.
Why do you say reaction 2? Because an aldehyde is becoming a ketone or vice versa? Because an aldehyde is becoming a ketone or vice versa?
Yeah.
And that's exactly what's happening here. And why was this important? Oh yes, because from an evolutionary standpoint, these 2 processes were probably related to each other. Eventually they separated, but they were initially the same thing. Collins says this reaction is also the same, and he's not right about that. It is very similar to another reaction that's on your cheat sheet. Obviously, it must be some kind of isomerization, and this one actually involves phosphates moving around. I have not talked about one of the molecules involved in this process. Does anybody recognize on your cheat sheets what reaction is identical to reaction 8? This is 8, right, Colin? Yeah, okay. And don't say 8 because 8 is the same reaction. thinking it's reaction 14. Am I right? Those aren't the same. Oh, they're the same. A phosphate's moving from carbon 1 to 6. Here's moving from carbon 2 to 3. And remember, in this process, is this actual phosphate being moved up here? I said this, and you don't have to memorize this. This you might have to memorize in more advanced biochemistry or bioenergetics classes. Is this phosphate the same as this phosphate right here? No, the enzyme takes this phosphate off, puts its own phosphate on there, or vice versa. You make a 2,3-bisphosphoglycerate molecule. I'm done talking about that. I don't care about the mechanism at all right this very second. What I do care about is that these reactions right here are going to happen twice. I don't get 2-glyceraldehyde molecules produced directly, but I can easily isomerize this one this way. And how do you know from this slide that this is an easy reaction? Arrows are going in both directions. And they're going in both directions here, and here, and here, and here. And this one is not going in both directions, because this is not a very reversible reaction. All reactions are reversible. All reactions that are catalyzed by an enzyme are absolutely reversible, because again, enzymes don't make reactions happen. They make a reaction reach equilibrium, and what's happening in equilibrium? The reverse reactions are occurring at equal rates. The problem is not that the enzymes can't catalyze a reverse reaction. The problem is that the conditions are never such that you actually reach equilibrium. And why would that be? Again, I know some of you are thinking, oh, it must be some magical process. It's like, no, it's really, really easy. In order for this reaction to reach equilibrium, I have to have the concentration of this times this, divided by this times this, equal the equilibrium constant, which unfortunately is a pretty gosh darn big number, because these guys are a lot more stable than these two guys are. I know you're saying ATP molecules aren't that stable. Yes, but compared to this one, they are. Thus, at equilibrium, I got lots of these and not much of these guys. In reality, I hardly have any of this guy lying around. Why? Because it's always going off in some other reaction. There are a bunch of things that you can do to pyruvate molecules, including making them back into glucose molecules, which we'll talk about in just a second. All right? I go from here to here, I get 2 ATPs. If I make this into this, then I go through this reaction, 7 reactions twice, thus I get 4 ATPs back. If I spent 2 to get into the process in the first place, then I get 4 back. That's a net of 2. And who's memorized that number before in another class? A couple of you have, I'm sure. All right? Do you see that on today's quiz, which you should be completing right now, that this information doesn't really help you all that much? Why not? Because I'm not asking you about glucose molecules going through pyruvate. I'm asking you about fructose 1,6-bisphosphate, or fructose 6-phosphate, or glucose 6-phosphate, or hey, on the tests and on quizzes next week later on, I could ask you about fructose glucose 1-phosphate. And all of these are worth a different number of ATPs than glucose is. And the reason I do this mainly is so that you guys stop just memorizing numbers and saying, where am I getting these things from? All right? I think a number of you did do that over the weekend and over the last couple of days. How many net ATPs is a glucose 1-phosphate worth when it's converted to pyruvate molecules directly in processes involving glycolysis? You all know I can make the glucose 1-phosphate molecule into glucose 6-phosphate. It's on my cheat sheet. It's sitting right there. Very reversible reaction. So how many ATPs is that worth? Not 0.
Would it be 3 then?
The net answer would be 3. And why isn't it 2? Or why isn't it 4?
Well, because there's already one phosphate group on.
There's already a phosphate group on it. And if I can make that phosphate into glucose 6-phosphate, then I don't have to use an ATP to make glucose 6-phosphate. So I'm only down 1 when I go from fructose 6-phosphate to fructose 1,6-bisphosphate. I'm still going to get the 4 ATPs back. How many net ATPs is a glyceraldehyde 3-phosphate molecule worth? 2, as is dihydroxyacetone phosphate, as is 1,3-bisphosphoglycerate. This is worth 1 going on to pyruvate. How are we supposed to keep all those numbers straight? By memorizing them? No. You look at your cheat sheets there. If I start here, I go down to this, here's how many I get back, here's how many I put into this, all right? That's why you want that cheat sheet, so you can say, here I'm losing 1, here I'm losing 1. If I start here, I go down to pyruvate molecules, and I just get those 2 produced directly in the glycolysis process. That's an important term right there that I'll make several different distinctions about as we go along, all right? Once I've made that pyruvate, oh yeah, this is important. Guys, this whole process happens maybe in some different places, but in the cytoplasm in general, all right? It's some separations of some of the different bodies and some organisms. But the main thing is that's not happening inside the mitochondria. When I make glucose into pyruvate, when I make pyruvate into lactate, which is not part of glycolysis, all of this stuff is happening outside the mitochondria, okay? Why is that important? The remainder of the oxidation of a glucose molecule, only partial oxidation through glycolysis, is going to occur primarily in the Krebs cycle. The Krebs cycle takes place inside the mitochondria. This is why you learned about this seriously in 8th grade, as the mitochondria are the powerhouse of the cell, because you're doing all the big oxidation reactions, fatty acid oxidation, electron transport, and all the Krebs cycle reactions inside the mitochondria, not outside. Glycolysis takes place outside, a much more anaerobic set of conditions, which again, somebody may have heard about in evolutionary terms. Presumably, mitochondria evolved from some free-living bacteria that were much more aerobic than the external part of the mitochondria, outside the mitochondria, parts of cells, which will be more anaerobic types of processes. We'll get to in just a little bit, okay? There are only 2 oxidations that occur in glycolysis completely, from glucose or fructose or any of those 6-carbon molecules, and they occur in that reaction we just saw before. Well, what reaction number is the oxidation reaction in glycolysis on your cheat sheets? What do you got, Colin?
Might have mistaken it for a different.
What do you think? What do you think?
I was thinking either number 6 or, actually no, it's only number 6.
It's only number 6, and how do you know that? It's the only one that involves an oxidizing agent, which is what? What? Did somebody just say NAD?
Yeah.
You have to say NAD plus, right? NAD plus, I'll go along with that part. Hey, real fast, I don't care about it right now, but we will talk about it when we talk about Krebs cycle. What does NAD or NAD plus or NADH, what does the NAD stand for? I guess maybe a better question. Of the 3 letters, which 2 of them should you be able to draw? I'm not talking about the letters, but the actual structure that they refer to. Why? Because that stands for adenosine or adenosine diphosphate dinucleotide, kind of means the same thing, all right? 2 phosphates, a sugar ring, a ribose ring. Why ribose and not deoxyribose? Would that change the energy of the molecule? Not for the phosphate part, but is adenosine diphosphate or deoxyadenosine diphosphate more energetic, it would take more energy to make in the first place? The deoxy one, right? So we don't do that. We're not worried about that in this situation because we're not going to be making any change out of these molecules. We're just interested in the phosphate parts of this. What does the N stand for? And more importantly, where do we get that N from? By the way, Kai, I meant to tell you, you look really good today. I'm not used to seeing you in anything besides sweats and ultimate frisbee stuff. Sorry. Huh? Yeah, did you feel like you were getting reamed?
Yeah.
That
I
wasn't
did so
really that bad.
well before you got there. And then
Actually, it wasn't that bad. But then I don't know what they were talking about. In some cases, what letter is it? Yeah. Oh, and when you pull out the denaturation, it's like, oh.
It was just the first one that came to my head, and I was like,
And unfortunately,
it's
you
not
did
right.
kind of rectify things right there, and I think that kind of went a long way to making things okay. Anyway, we'll worry about that. This is later on. What does the N stand for? Kylie, do you know? Some kind of a mid is actually a good answer. Well, that's not really all that important for the reactivity.
It's like
Nicotinamide, nicotinamide. What is the
said it like 3 times.
socially acceptable name for nicotinamide? It's not nicotinamide because the nicotine part is bad. It's not really that bad. Anybody know how you get nicotinamide? You do not really make it. Nobody has any idea about this. I'm interested. Who has taken some nutrition classes? Who has had some nutrition in your biology class? You probably haven't had it in your chemistry class.
Vitamins?
It is a B vitamin, but I don't like using those letters, all right? Do you know which B vitamin you are talking about? And don't say a number. I don't like that even more.
Thiamine?
Thiamine is not the correct answer. How do you get an N out of
Riboflavin.
thiamine? Does anybody know what B vitamin does start with the letter N?
Nice.
Niacin is the correct answer, and niacin used to be called nicotinamide or nicotinol, but they changed it to niacin because nicotine was bad, so we didn't like that name as much anymore. I have to eat enough of that to make all the NAD plus that I need. Guys, this is the important part about this conversation. I am not making NAD plus or NADH when I am actually making glycolysis or the Krebs cycle take place, all right? What I'm doing is converting those NAD pluses to NADH or vice versa. I have in my cells right now a certain pool of nicotinamide molecules in either the H or plus form. The adenosine part is just a marker for it. It's not involved in the reactions at all. Those of you who heard of NADP plus, NADPH, same business end of the molecule. The phosphate that makes it different from NADH is not attached to the nicotinamide part. It's someplace else on the molecule. Not important. The business end is the nicotinamide molecule, and when I talk about making NADHs, it's not making them from scratch. Even NAD pluses, it's also going to be when I'm making NAD pluses, I'm not making them from scratch, I'm making them from NADHs. When I'm out doing anything, athletic, thinking, whatever it is, I am not making bunches of new nicotinamide adenine dinucleotide molecules. What I'm doing is converting them from one form to the other in your cells. Mitochondria, outside the mitochondria, you have a pool of these molecules. I am stressing this right now because glycolysis inside you is never going to stop because you're out of glucose molecules. Some of you say, but I used up all my glycogen stores, then you're going to take proteins and make them into glucose molecules. Or you're going to take something else and make them into glucose molecules, not fats. The reason glycolysis stops in you is never because you're out of glucose molecules. You can stop eating, period. Not eat anything whatsoever. And 5 weeks later on, you still have glucose molecules floating on your bloodstream. Your amount of protein will be down a long ways, and you'll make some of those amino acids into glucose molecules, but you will not run out of glucose molecules. Later on today, if you go out and play soccer, go out and play football, go out and play basketball, go out and do your running or sprinting of any sort, you're going to run out of glycolysis taking place really, really quickly. But not because the glucose is gone, because all the NAD plus that's present in those cells that are doing all this exercising are depleted. They're broken apart. No, they're all in the NADH form. And I can't use NADH as an oxidizing agent. It's not. The part of NADH that's been reduced is the part that I use to oxidize other things. And if it's already been reduced, it already has the electrons, as Jess was now pointing out. I can't give it any more electrons. In order for me to get this glycolysis process going again, what I need is glucose. No, I'm not out of glucose. I gotta get the NAD pluses back. While you are sitting here, Harris, Madison, how are you getting those NAD pluses back? You are not athletically involved right now. You're sitting here, and how are those NADHs that are being produced in glycolysis being converted back to NAD pluses?
Aren't they going to be oxidized
Yes. By what process? In you right now, Colin.
from the formation of lactate from
No.
citric acid?
No.
Formation of oxygen.
Not from the formation of oxygen.
When you oxidize, form oxygen.
We don't do that.
That's the reason.
That's if you're a plant, you do that during photosynthesis. You're going to use that oxygen to get the NADH back. No, not CO2. You're going to form water. That's okay. So make sure you understand, you don't make oxygen. That's why we have to keep breathing, because we can't make oxygen that way, right? Where is that oxygen being used to regenerate NAD plus molecules? Electron transport chain, which is where again, Fonda? It's way inside the mitochondria. This is a problem. It is not a problem for you right now. You can easily get the few NADHs that are being produced in those cells that are doing some work inside the mitochondria. You don't get the NADHs indirectly. They transfer to something else. We'll talk about the shuttles that get them inside. But they're going to end up getting put in the electron transport chain, go to oxygen, make your water molecules, and everybody's happy about this. When you go out later on, what are you, a sprinter? Right? When you go out, especially a sprint and long jump, all right, you're going to be using those glucose molecules way too fast. Generating the NADHs way faster, and you can get them inside the mitochondria to make that electron transport chain. Then I have to have some way of getting those NAD pluses back a lot more quickly, and that is going to be involved. That's what's going to be involved in the formation of lactate. All right. What happens to a pyruvate molecule once you've made it in the process that I just described as glycolysis depends upon whether I need more energy and whether I have enough oxygen to keep the electron transport chain going, as Bonner just asked, or just suggested, all right? If I do need more energy and I have oxygen, what's going to happen to this pyruvate molecule is it's going to become the acetyl portion of coenzyme A. And hey, water on a limb. What does the A stand for in coenzyme A? Adenosine diphosphate. What does the coenzyme A part stand for? Jessica, do you have any idea? You open your mouth like you're going to say something. And...
No, he's stepping on a cough drop.
Okay. Okay. Other vitamins are involved, not nicotinamide in this case, not thiamine either. Talking about things like pantothenic acid and parts of amino acids. I'll show it to you later on when we talk about the Krebs cycle. In order for things to go this way and further oxidation, I have to absolutely have oxygen present. If I were a yeast cell, I would go this way when I don't have any oxygen present and I want to make energy. Guys, look at this. We don't go this way. All right? I've had people say, oh, yeah, you make pyruvate to ethanol. No, yeast cells do that. Other organisms that ferment pyruvate to ethanol will make ethanol. Why are they doing that? I can see some of you saying, to make people who work for Anheuser-Busch and bars much more wealthy than they are already. That's probably true. That's not why the reason that they do it. Why do yeast cells make pyruvate into ethanol? The answer is on the screen, folks.
Further reduce, actually no, is it, no, oxidize.
Oxidize what?
NADH to NAD plus.
And why would they go about doing that?
Think of it as a way of having NAD plus continue their glycolysis cycle.
But I'm thinking of that. In a way to keep my glycolysis. Why don't I just send them to the electron transport chain?
They don't have that.
Yeast cells don't have electron transport chains?
Actually,
Everybody's got some kind of electron transport chain. Right? Not everybody has the same one that we have. And yeast is a little bit different from ours. Not that different. All right? Some of the proteins involved in electron transport chain in yeast cells, very, 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 very close related to ours. All right? Doing the exact same jobs. Every eukaryotic organism has an electron transport chain very similar to ours. All right? Every prokaryotic organism has some kind of electron transport chain. Some of them are very, very different than ours, all right? Some plants and other vegetation type of organisms have other electron transport chains that are quite a bit different from ours, but also some of them are still similar to ours too. Why don't yeast cells just use those electron transport chains? What do I have to have for my electron transport chain to work?
Oxygen. That was, isn't it? Oxygen?
Oxygen is the correct answer. How does Anheuser-Busch get yeast cells to make ethanol? They feed them a bunch of glucose molecules, and then they choke them. They say, hey, you don't get any more oxygen right now. In order to get the NAD pluses back to keep their little glycolysis taking place, they'll go this way. Again, folks, we don't do that. Some of you might, and I've heard this happening especially with diabetics, get an infection where an organism will do this, and under more anaerobic conditions, they suddenly start getting ethanol produced because the bacteria that are in them, or some kind of yeast cells that are in them, will actually produce the ethanol molecules. This happened, I guess, I've heard one anecdotal story about something during World War II, where a guy was over in Japan, a GI was over in Japan, and was treated in a hospital for drinking. They just assumed he's a GI out on a bender. He said, no, I don't drink anything at all. Alcohol is like really high alcohol concentrations in his blood, because some bacteria was taking the glucose molecules and under anaerobic conditions, making ethanol. And who was in my office was thinking about this? Was Jackson or Mark, somebody was in there. If you could do this, and you recognize that you could just eat a bunch of sweet stuff, jump around for a little bit, hold your breath, and then you suddenly start making more ethanol to replace the NAD pluses that are being used up as these molecules of glucose are being converted to pyruvate. Inside us, under normal circumstances, when we don't have enough oxygen, we make this molecule. And you guys should all know right now, because you know what pyruvate looks like, exactly what lactate looks like. Some of you say, well, if I look at my pyruvate molecule, there's two things that could be reduced on that. And of those two things, which one clearly must it be if you're making lactate? Who knows how many carbons pyruvate has? If you know the answer to this question, please raise your hand up high right now. Christy, you know how many carbons pyruvate has? Who knows what at least one of those carbons is? What do you got, Wasim?
One of the carbons is another group.
You know that. That's actually pretty good. From the name pyruvate, what does one of those carbons have to be? The 8 part tells you this. I think you told me this the other day, didn't you? Melanie, what you got? Lactate, pyruvate, and help. A carboxylic acid group, correct? Pyruvate has 3 carbons. What's the non-methyl, non-acetic acid? Not